Okay.
Maybe you should wait a few more minutes.
Yeah. So Emily, you'll let me know if anyone on Zoom has any. Okay. Right. And then I don't remember so many pieces of technology.
Yeah.
I think because of the Zoom, there's one more thing here. Oh,
But I mean, because this goes through here.
yeah.
Emily, does it need the microphone necessarily, or is
I think
better?
I think
Just
it's more
for the room.
people in the
Okay.
room. Okay. Yeah. And then you know how to make this disappear? It's like hide something.
Did you disconnect the projector onto this?
Oh, okay. That is new.
Did it unplug accidentally?
Yeah.
Do you want to unplug it and replug it? Okay, there we go.
Good.
And it does have this on there. So you might just want to click this.
So I want to share screen. Okay, so now this is sharing. I want to click on this. Okay, and then
And you can hide that if you want. If you click more, and then you should
hide floating meeting
Yes.
controls. Okay, good.
Yeah, and then I'm going to turn off the stage board lights once you start presenting, so it's easier to see your.
And then this one, I should turn it on.
And then you should be good. Okay, I'm gonna turn
Okay,
off the stage.
can people hear me? Yes.
Okay.
There you
Yeah.
go. Okay.
I don't know whether this. I think so. I think so. Actually.
Did you hear Tzer Han in the back? Okay.
Okay, great.
Okay. So I guess can we start at this?
Maybe I don't know. Do you want to wait
Yeah.
a couple of minutes or I don't know?
Yeah, it's
You're the
fine.
host.
I mean,
You're the
Emily is the boss. So it's a great pleasure to have Tzer Han Tan here. Welcome back to the qBio seminar. Tzer Han is a new colleague at in our department of physics. He did his PhD at MIT, then a short postdoc at Harvard, and less short at the Max Planck. And Tzer Han has done seminal experiments to understand symmetry breaking and organization in living matter. And today he's going to talk about life as an emergent phenomenon. Thank you, Tzer Han.
Okay. Thank you, Mattia, for the kind introduction. I'm very excited to be here, and I hope to get to know all of you in the coming months and years. And so for today, I thought I would share with you my perspective of how to see life as an emergent phenomenon, how we can use the framework of active matter to describe it, and also and then I'll share two short stories on my recent work. So when we look at living systems, I think one of the things that we realize is that it's typically highly complex, but at the same time, there's also some level of regularity both in space and time. And invariably, this regularity actually comes from collective behavior from microscopic components. So this happens across all different scales of biology. So for example, in a cell, there are these mitotic spindle structures responsible for chromosome segregations. And these structures, these intricate structures, is actually composed of microtubule filaments and kinesin motors and hundreds of others regulatory proteins. But the collective behaviors of microtubules underlies the formation of these structures that allows cells to divide properly in all major cell types. At the tissue level, increasingly, people are realizing that of course, tissue flow is important for morphogenesis process. As you can see here, this process is underlined by the coordinated motions of thousands of cells working together to ensure robust development. And even at the larger macroscopic scales, we see phenomena such as this bird flocks, where thousands of starlings actually line up with their neighbors to form these large-scale structures. And what is remarkable about this is that over the past 2 decades, the active matter community has come up with a framework, the general framework that are able to elucidate a lot of the mechanisms that underlie the collective behaviors across these different scales. So let me first explain what I mean by active matter. Active matter essentially are systems which are composed of cell-driven components which are able to interact with their neighbors, and that leads to organized macroscopic states. So this self-driven components they can come in different shapes and sizes. So for example, they can be isotropic or they can be elongated, so that when they align with their neighbors, there's kind of a nematic interactions. And on top of that, you can also get a more complex symmetry in their interactions. So one of the most common example is when if you have polar order, meaning to say on top of this elongated structure, you can imagine that each of these little components has an additional arrows, and these arrows wants to align with their neighbors in the same direction. And depending on the kind of particles and types of interactions that you have, we can actually, there are many intriguing non-equilibrium phenomena that can arise, such as non-equilibrium order disorder transitions, pattern formation on mesoscopic scales, as well as leading to unusual mechanical and rheological properties. A very prominent biological context in which this framework has been very successful is in tissue morphogenesis. So increasingly, people have shown that we can think of cells as having a nematic and polar order, and the arrangement of this nematic and polar order can explain a lot of the macroscopic phenomena that we see in tissue morphogenesis. One of which is this topological defects. So topological defects are basically special points in polar or nematic field. I'll explain that in a bit more detail later. But recent work has shown that topological defects in polar or nematic field can actually access organization center in tissue morphogenesis. So for example, in this picture, this is a picture of Hydra, and what is labeled here are basically actin filaments. The group of Kinnera-Caron has shown that defects in this actin nematic alignment field can actually access points in which the future head and the future arm will actually grow during Hydra regeneration. So on the right here is a picture of tissue culture as a muscle cell. And what is shown here is that the defects in the nematic field actually correlates with points of differentiation. Other contexts in which this active matter framework is useful is that the group of Ojera-Campas has shown that the body axis elongation of zebrafish actually is a type of a jamming transition. The cells in the progenitor region, when they move to form this pre-somatic mesodermal segments, they actually undergo this fluid to solid jamming transition. So these are examples at the tissue level. At the cellular level, we can also, at the cellular and subcellular level, especially in the cytoskeleton systems, the framework of active matter is also very powerful. So this is the mitotic spindle that I was showing you earlier. And what people are able to do is that they were able to purify this microtubule and kinesin motor proteins. And when you feed this system, this reconstituted system with ATP, they are able to generate this type of spontaneous chaotic flow mixtures in vitro. And the advantage of bringing cytoskeletal system into an in vitro context is then that now we are able to more easily manipulate boundary conditions and initial conditions. So for example, the group of Zvonimir Dodzik has been able to encapsulate some of this in vesicles so that under this dynamical flows, you essentially get dynamic shape changes of vesicle that is controlled by points of topological defects. And if they have purify enough of this protein to occupy this 3D volume, they found that you actually get disclination loops with very interesting topological structure. So I think the framework of active matter is very powerful because it allows us to try to understand how non-equilibrium forces drive spatial temporal organizations in living matter. And it also raises interesting questions on how biology regulation in turns make use of this self-organizing capacity to make structure and function. And so in the past few years, I've worked on a couple of projects ranging from the organelle scale to the colony scale, addressing questions such as how stress patterns are generated in ectomyosin cytoskeleton, how pattern formation in row signaling proteins are formed, collective cell migrations, as well as hydrodynamic flows. But today, I would like to just focus on two short stories. The first one is on living chiral crystals, where we make use of C-star embryos to investigate how chiral interactions can lead to new emergent behavior in active matter system. And then in the second part, I will talk about how a story on collective cell migrations on a confined spherical surface and how that can lead to a chiral symmetry breaking. Okay, and I should mention that feel free to interrupt at any point with questions or clarifications because I realize that people might have a different background here. Okay, so let me first talk about jump right into this first story of living chiral crystal. The system that we are looking at is composed of this starfish embryo. These are about 300 micron in length and 200 micron in diameter. And on the surface of these embryos, they are basically a couplet of cilia, which are these structures that can beat in synchronous fashion to generate hydrodynamic flow. And of course, this flow is important for motility of these embryos. But the hydrodynamic field is such that when these embryos swim near an air-water interface, they actually starts to form a stable bound state such that they are aligned perpendicular to the interface. And again, because of the hydrodynamic flow, we start to have the embryos begin to attract their neighbors on the surface. So here is a movie where we are looking from the top. So each of this little circle right here is an embryo. And what we found was that if you have enough of these embryos in, like, say, a petri dish, they can spontaneously self-organize, self-assemble into these large crystalline structures, which shows a global rotation. So notice the time scale and the length scale here. So we are talking about a 2 millimeter scale bar. So these are really, you can see this crystal by eye. And the time scale here is of the order hours. So the crystals self-assemble, they sort of persist for up to 30-ish hours before they dissolve away. So these crystals have all the properties that you would expect of a 2D crystals. You get 5-folded and 7-folded dislocation, defects, disclination lines. But what makes this an interesting system is the fact that all of these embryos are actually rotating in a clockwise fashion. So if you look at this video right here, each of these individual embryos are actually rotating in a clockwise fashion. And so this system has a broken chiral symmetry at the particle level, and we are interested in asking what kind of new emergent phenomena do we get once you have this chiral interaction. So of course, as you can see, this is a very this system has very rich phenomenology. Yes.
This is just suspended.
This is just suspended in a solution. So you take a petri dish, fill some seawater, collect thousands of embryos. They start swimming to the air-water interface.
Oh.
So we are imaging. So we are imaging at the 2D slice at the air-water interface.
And they'll collect a few data.
Yes. So because they form stably, so they form a stable bound state near the interface, so they swim around, and then when they see an interface, they get stuck there. So essentially, it forms like a sink that sucks in all the embryos. Yeah. But then, as you can see, once the crystals are formed, there are still some embryos that are swimming behind. So that's the bulk. Yeah.
And is it always clockwise or is it just always in the same direction?
It is always clockwise. So of course, when you have like embryos that has some developmental defects, they do weird things, but then on average, they are all clockwise. Yeah.
Is there some buoyancy that like points them towards the surface, or like, how does it find the surface?
Yeah, so sometimes this phenomena can form at the bottom. So there's the buoyancy doesn't play a role. In fact, if you kill the activity of the cilia, these embryos will sink down. So they are heavier. Yeah.
So is there some developmental stage when you start rotating, or is some external signal which
So it's the developmental state. So they start to form at about 24 hours post fertilization, and then they dissolve at about 60-ish hours. I will mention that in a bit more details later. Yeah. Yes, they are all rotating even they are not assembled. So if you look at single embryos trajectory in the bulk, they actually do a corkscrew motion, because on top of propulsion, there's a rotation, and there's a difference in the propulsion and the rotation axis, and that allows them to make a corkscrew motion. And that is very common in a lot of marine larvae. Yeah. I'll get to that in a moment. The short answer is we see that in other species, but we don't know whether they perform any specific function yet. Yeah.
So all these embryos are related, right?
They are from the same species.
Right. I was wondering if there's if you put in like a whole bunch of different embryos from all bunch of different
Right.
of the same one, do they only form with games?
As far as we know, there's no self. They don't know whether they are brothers or sisters. Yeah. But that's a interesting question. Okay. Cool. So for today, I would like to focus on sort of 3 questions. One is the self-assembly process. How does it self-assemble into this crystalline structure? What kind of new emergent dynamics do we get from this chiral dynamics? And then, why the crystal undergo a dissolution process. So for the self-assembly process, the first thing that we tried to quantify was the hydrodynamics around the embryos. So we put beads around these rotating embryos, and what we found was that the PIV trajectory basically shows a net inflow towards the embryo, and then of course, and once they are near the surface, this fluid is actually expelled down into the bulk of the system. And what we've showed is that this flow field is well described by a Stokes-Lut that is generated by a single point force, and this accounts for basically the attractive interactions once the embryos go to the surface. But then, of course, what is more important is what happens when you have a pair of embryos and how do they interact with their neighbors. So here we have a set of similar experiments. We look at a pair of embryos. We put beads around the fluid, and then what we found was that the 2 embryos rotate clockwise. They start to attract each other because of the Stokes-Lut flow, and because of the rotation, they start performing this orbital type motion. Yeah. So this inward flow is in the top plane. So the surface of the world. Exactly. So it's if you image further down, it actually goes up. It's deep beneath the. Right. So this so what we because of this rotational motion, they start forming this orbital motion when a pair of embryos come together. And essentially, you can think of 2 types of interactions involved. One is the transverse force interactions, where because of the rotation, this embryo is imposing a perpendicular force on the other on its neighbor, and this essentially explains why they start to orbit around each other. But on top of that, there's also a torque exchange due to a lubrication effect between the embryos. And if you notice, because they all rotate in a clockwise fashion, at the point of contact, they are actually moving in the opposite directions. And this has a net effect of both embryos slowing down their neighbors simultaneously. And so based on this description, our theory collaborator, Alex Meeker, came out with a sort of a minimal model to try to explain the chiral crystal formation. We treat these embryos as 2D disk, and then this equation of motion basically says that there's a Stokes attraction, there's a short-range steric repulsion, and then there's this transverse force coming from their neighbors that is due to this rotation of the embryos. And this chiral force will depend on both the omega, which is the rotation frequency of the embryo. And to close the model, we say that the spinning frequency of each embryo has some base free rotation speed minus the neighboring torque that is that basically comes from this lubrication effect that we were talking about earlier. And using this simple model, what we are able to show is that qualitatively, the simulation can recapitulate the dynamics that we see in our experiments. And at the same time, we also get a good agreement when we measure embryo spinning frequency as a function of number of neighbors. So in small clusters, for example, this guy has 2 neighbors, this guy has 3 neighbors. And because of the more neighbors you have, the slower you get because of lubrication effect. And on top of that, we are also able to get good agreement between the cluster rotation frequency, so this macroscopic cluster rotation as a function of number of embryos. So that's all I have to say about the self-assembly process. In the next part, yes.
So what are the other bits? Can you slide? So what are the parameters that you are keeping from data?
Okay, so the Stokes attraction we can actually approximate from the buoyancy force. So by killing the embryo, we find that the embryo is sitting down. So we just assume that the propulsion force needed to allow the embryo to stay floated at the air-water interface is the point force that generates this Stokes. Then we can measure, of course, the rotation rate of this individual. So we have this free rotation speed. And I don't think there's no free parameters in the model, I think is the bottom line, that we are able to, through independent measurements, come up with terms here and here, but we can talk details. Yeah. Okay. So in the second part of the story, I want to talk about the emergent dynamics that come from the chiral rotation of this embryo. But before that, I wanted to let me set up the stage by talking a little bit about linear elasticity theory. So if you have a piece of 2D isotropic passive material, typically we can characterize the elasticity property by 2 moduli: the B, which is the bulk modulus, and mu, which is the shear modulus that tells you how much force you need to squeeze the material and how much force you need to shear the material to get a certain shear response. And these 2 moduli are typically linear with respect to the kind of longitudinal interactions that you have with respect in your atoms. But in our system, we actually have, on top of longitudinal forces, we also have transverse forces. And what this paper has shown is that once we have this additional transverse interaction, we actually get 2 additional modulus in this elastic moduli. So here we have A, which couples compression to internal torque density, meaning to say if I squeeze a piece of material, you can actually get a rotational torque in the middle of the material. And then there's this antisymmetric K0 shear coupling, which basically rotates the shear response of the material. And these are these are called the odd moduli because by virtue of being off diagonal, it means that our system must be out of equilibrium. And what this paper has shown is that this material is able to support waves even when then, and that's because the constituent particles is consuming energy to overcome this damping. So what we observe in our experiment is that actually there are sometimes we can get the crystals to vibrate at a certain frequency. So here is a video of this crystal, and right at about this point, you can see that there's, even by eye, you can see that there's this jiggling motion going on at the crystal and stage. And in order to quantify this vibration of the crystal better, what we did was that we were able to track this individual embryos and then compute the displacement vector. So here, the tiny black arrows, each of these tiny black arrows is the displacement vector of a single embryo, and the color code tells you the angle of this arrow with respect to the x-axis. And what you see here is that during this vibrational mode, there are these chiral waves that propagates. There's this vibrational states that propagates across the crystals, and then the displacement vector typically make a circular motion trajectory. One thing that I want to point out is that the period of this displacement is of the order 240 seconds. So we're talking about minute timescales. So given that this is a overdamped system, it means that the vibrational modes that we see here must come from the non-equilibrium activity of the system. And inspired by the paper from Schneider et al., we also compute the different strains. So from the displacement field, we can compute the strains as a function of time. And what we found is that if you look at the tiny piece of this crystal and you compute the curl, the divergence, the shear one, and the shear two as a function of time, they basically perform this anti-clockwise cycle when the wave is passing through, which is consistent with the signature of what was reported in this paper. So we actually did some additional work to try to measure, to try to infer the odd elastic modulus by using, yes.
What are you looking at those values? What is X, Y, and what color does the color stand for?
Okay, you mean this plots right here? Yeah, so these black arrows are the displacement vector. Yes, so these are the displacement vector. And from this displacement vector, if I take the gradient of it, I can get the strain. So from the strain, I can take a linear combination of the strains to define the curl, divergence, shear one, and shear two. And because this is a movie, I can do it as a function of time. So basically, if I plot it in this space, it will be some trajectory that moves in this space. And then the way that I generate this plot is that I sort of discretize this phase space, and then the intensity. So the yellow and the blue, the intensity is basically the density of states in this space, and then the arrows are the transition probabilities in the states. So it's just a way for us to try to quantify, to detect cycles in the strain space.
Which is for a single spot. If there is squared for a single spot in space, the trajectory reflects the time of how it moves in the different states.
Yes, so this trajectory describes the strain dynamics in a small piece of material. So in principle, I can do this for every point in space. So I will get like lots and lots of this plot. Yeah.
In this movie, do you like how, because in the video the whole thing seems to rotate?
Yeah.
And did you subtract out the
yes
rotation?
So we actually go into the co-moving frame of the crystal.
And it moves as a rigid body.
It moves as a rigid body. Yes. So it does move as a rigid body.
the two plots and curl and divergence of shear and shear. It's the same information, just different.
They're actually different information because displacement, well, let's say you have dx dy, and then if we take the gradient of displacement, then you will have partial dx, partial x, partial dx, partial y. So you have 4 independent components, and then these are linear combinations of the 4 different components. Yeah. Yeah. Okay.
How exotic is odd plasticity, which you're still odd materials? People use for anything?
So people are trying to build odd materials. So odd viscosity has been around for a while. Like I think, okay, I think the quantum people know about odd viscosity, but odd elasticity is more, the theory of odd elasticity is still being developed. And then in terms of materials, there are, I want to say there are maybe at most 2-3 different synthetic systems within the past few years people have tried to build. And biological systems, I believe we are the first to show signatures of odd elasticity.
Is there any use for materials?
Yes, so all materials they are useful because they can redirect strains in some way. So there are papers from Corentin Coulet's group at Amsterdam which shows that you can make robots that can cycle uphill, or that you can make materials that can actually soften the impact, because if you have an impact coming this way, they can actually shear the response in some other direction. So it could yes, there's definitely once you have this exotic material properties, you can think of applications for that. Yeah. Okay. So that's all I have to say about the second part on our emergent dynamics. And so the last part I want to discuss is this dissolution process. So what you notice is that at about 38 hours, these crystals start to dissolve. And if you stare at the movie long enough, you'll notice that actually in the initial stage of the crystals, the embryos actually do not fluctuate less. But then as they move towards this dissolution phase, the embryos actually fluctuates more and more. And so in order to quantify this fluctuation, what we can do is that we can quantify this local bond orientational order parameter, which is defined here. But intuitively, one way to understand this is that the magnitude of this order parameter quantifies the local hexagonal order. It tells you how hexagonal locally this atom is. And then the imaginary part tells you the phase of the size 6. So basically, what is the angle this local hexagon is making with respect to some reference axis. So if we color code the embryos, if we compute this, color code the embryos as a function of time, what you will see is that initially the crystals has very little hotspots, meaning to say they are quite hexagonal. But then towards the end, there's more and more hotspots coming on. Turning on in the crystals. And this is reflected in the orientational order. Initially, most of the crystals has one color, meaning to say they have one crystal axis. But then afterwards, you see there's more of this cracks appearing in the crystals, and you get smaller and smaller pieces of crystal. And we can quantify this development of disorder more quantitatively using this probability time chymograph. So here we are looking at, so this axis is the magnitude of size 6. This horizontal axis is time. So each of these slides is a probability distribution of the magnitude of size 6. So initially you see that there's a tight bright band near 1, meaning to say most of the atoms in the crystals have high hexagonal order. But then towards the end, you can see that the distribution spreads out towards the lower end, where you get more and more hotspots. If you take the average of this chymograph as a function of time, if you plot mean size 6 as a function of time, you can clearly see that there's a decrease in order. And one thing I want to mention is that we are not changing any external parameter. So I'm not tuning the temperature or viscosity of the system. Everything happens because of the natural development of the embryo. So I think there was a question earlier that as a function of time, these embryos, of course, are also continuously developing. So this is how they look like during the course of our experiments. initially they look nice and symmetric, roughly with very high azimuthal symmetry, but then eventually they develop more and more structures, and their hydrodynamic flow fields change as well. And in order to sort of quantify some of this asymmetry that develops during this development, one thing that we did was that we just look at the ellipticity of the embryos when you look from the top. So these are the top view of the crystal, and you can see that initially they are nice and circular, but then as a function of time towards the end, they become more and more elliptical. So that actually introduced a source of noise that leads to the dissolution. And another thing is that if you look at the boundary, the embryos at the boundary, because of this shape anisotropy, when they are nice and isotropic, it's easy for them to remain perpendicular. But then once there's shape anisotropy, it's easier to topple some of them over, and then they start swimming away. And that's also contributing towards the dissolution of the crystal. So right. So in this part of my talk, I've shown you how hydrodynamic interaction leads to self-assembly of the crystal, what chiral interaction does to the emergent dynamics of the system, as well as how development that breaks symmetry at the particle level leads to dissolution on the crystal. And I think there are actually a lot more that we can do with the system. Yeah.
So it looks like they assemble, but then they don't rotate right away, right? It takes time before they start to rotate.
Yeah. So that is something that we don't understand. And it's very, sometimes they start rotate right away. Sometimes they seem to get stuck before they start rotating. There are instances where the crystals are rotating and then they get stuck and then they continue rotating. So we don't know what's going on.
The embryos are rotating.
The embryos are rotating. Yeah.
Maybe I missed it, but is the internal dynamics, the developmental dynamics, is it independent of what is happening outside?
Yes. Okay. Right. So if I do this in a crystal, and if I measure embryos that are freely swimming, afterwards they are developing normally.
So why are they do they self-organize in their natural habitat
in the open ocean, we don't think so, because these are rather fragile. If I nudge at the petri dish, they break apart. But if you imagine these embryos developing in tide pools, I think there are environments that they might do this. Which actually, maybe I'll answer one question and then I'll come back to that.
Considering the dissolution to a local effect, like each cell locally break away from its statements, or is there like a macroscopic scenario?
So it's more like an evaporation. Think of it as an evaporation process. The embryos are leaving from the boundaries, and so there are two things. One thing is that because you see that there are cracks that are happening, but then most of the embryos are leaving. The crystals leave from the boundary. That's it. Makes sense. So if I can somehow confine them, grow dense enough crystals with very defined boundary conditions so that they don't swim away, I think we can push the system further to stay in the crystal state. Yeah.
Well, you started this rigid body rotation.
Yeah.
Do the spinning velocity of single embryo, is it the same as before they were?
No, they slow down. They become slower.
In terms of energetics, have you looked if there is some sort of conservation of physical kinetic energy?
It's hard to think about conservation because we also have the fluid around. Yeah, there's friction, and then some energy is being dissipated in the hydrodynamics, and then
but just in general scale. So if they slow down, because maybe they're giving a bit of energy, the translational motion of the rigid body.
Yes, I think there's part of the energy that transfers to the big rotation, but then there are also others that are being dissipated. I think that's sort of how we understand it.
What's the translational cell division?
No, so we are talking about a single embryo. Cell division happens within the embryo. Okay. So I guess what you're asking is how many cells here versus how many cells here. at least double. So I think at this point, there are probably of the order 1,000 cells, and then here a few thousands. Yeah.
Yeah, attractive forms when you model when you flow the input theory, like can you write down a function of the attractive force as a function of the shape and as a function of the particle? Maybe like what you form a more
we can't really write it down as a function of shape anisotropy, but it's something that we can measure. I can take the embryos at 10, 15, 20, 30 hours and then measure the net inflow, fit it to the Stokes, and then that gives me the attractive force. Sorry. Is there a limit? No. At least as far as you know, if you have dense enough embryos and big enough surface, they can keep growing. Yeah.
The embryos pretty much the same stage of development all the time.
Yes, they are pretty synchronized at these early stages. Okay, so maybe I have 2 more slides with this that might be of interest. So yeah, coming back to this question of, what is the function? One thing that we did was that we look at other species of marine larvae. So this is the species that we use. It's called Pateria minata. It's actually the West Coast species. This is the East Coast species, Asteria forbacea. This is a sea urchin, and then this is, so they are all echinoderms, close cousins of each other. This is an outgroup, Platinares, which is a sea annelid. But what we found was that all this, the commonality is that their embryos swim in the open ocean, have cilia that beats around, generate hydrodynamic flows. And what we found is that in all cases, at some developmental window, we are able to see some evidence of crystal formation. And so then, you can ask whether is it purely an epiphenomena or, or this. I think this observation suggests that maybe there are some other reasons that they do this that we don't yet understand. But from a active matter perspective, I think this is, oh yes.
I'm just curious. I know lots of zooplankton and smaller size creatures can have plasticity to increase in size when their predators are around. Could this be something to make them that a predator can't swallow them if they're stuck in the
Yes, I think that's one possibility. Yeah, but I don't know how you can show it. Yeah, it would be cool if we can show it, because then I can tell people that they actually do something. They use it for some reason. Yeah.
Yeah, I'm wondering if there's a gene that holds the flow like that. If you modify that gene, make it less part of the population can be anti-clockwise and mess up this synchronized.
Right. 2 slides. Okay, I'll talk about this in 2 slides. Right. So I think from at least from an active matter perspective, I think this is interesting because now we have this zoo of particles that we can play with different shapes and hydrodynamic fields, and we can make different chirocrystals with different material properties and study them. And one particular species that I'm very excited about is this Platinares. So this is a schematic of how they look like. They, instead of a couplet of cilia, they actually have cilia at the equator and some apical turf. And this video right here, they shows you that they do form a crystal stage, but then they are more dynamic. It's less, there's more neighbor exchange and merger and separation event. We have very preliminary measurements of their hydrodynamic flow fields. They are roughly dipole, which is qualitatively similar to what we see in sea stars. But what is exciting about this system is that these embryos also have eye spots, and which confers them phototactic ability. And so the hope is that we can use light to manipulate some aspect of their hydrodynamic flow field so that we can make crystals with some with many more ways to poke at them. And coming back to this question of chirality, I think one other open important question is, why do these embryos all rotate in a clockwise fashion? And I should also mention that in all the species that we have checked and other species that we have talked to people about, it seems like marine invertebrate larvae all rotates in a clockwise fashion. So there seems to be some conservation there. So they are on the surface, they are covered with a couple of cilia, but then as they develop, they actually forms more complex bands. These cilia are beating around, and if you are just focusing on this question of chiral rotation, then it means that, I mean, there are several possibilities, but it means that, I mean, the cilia must have some left right information. One possibility is that maybe this cilia is somehow nodal cilia, which means nodal cilia is this cilia that is involved in left right symmetry breaking in some higher level animals. And that, you know, because that the cilia is beating in a chiral fashion, regardless of how you arrange them, you get a net chiral rotation. The other possibility is, of course, that there's some extrinsic signaling along the developmental pathway that specifies a certain direction for the cilia to line up and be such that they all rotate in a chiral fashion. And beyond this, I think there are also interesting questions on how on synchronizations and topological defects of this cilia patterns. Metachronal waves has been studied a lot of times, but then how do this synchronization gets affected when you put them on a closed surface like spheres, where you force them to have topological defects? I think is a very interesting direction. And then I think it also goes back to the earlier ideas where perhaps these topological defects could be organizing centers of morphogenesis and dictate some of the properties of the hydrodynamic flows of this embryo. Okay, so that's all I have to say about this project. Maybe if you have any question. If not, yeah.
As they go through the individual embryos, does their intrinsic rate of spinning change substantially?
So for the stages that we measure, they are actually roughly the same.
Once it starts.
Yeah, but then the cilia structure actually changes a lot from a couple of cilia to ciliary bands. So I would say the readout of the rotation rate is maybe not as informative as the actual patterns and flow fields. And the flow fields you can actually measure quite easily, right? You put beads, you do PIV. Yeah. Okay. If not, I will want to move on to my next story, which actually is somewhat related to this cilia chirality of this spinning embryos. Because now instead of looking at spinning embryos, we are looking at spinning of a epithelial tissues that is confined on a spherical surface. So let me first introduce the system. So here we are growing pancreas progenitor cells, so pancreas stem cells in matrigel. And what happens is that when you do that, they can actually self-organize into this single epithelial layer tissue. So at about, once you see the cells at about day 4 and 5, they develop into this kind of structure where the apical site is facing inwards, the basal side faces outwards, and so the cells are crawling against the matrigel that is facing outwards. And then the inner side is actually a fluid filled lumen. Okay, and what my experimental collaborator Irena realized was that if you image some of this as a function of time, you can see that this tissue actually performs a rotation in culture. And the time scale we're talking about is tens of hours. So this guy spins around, and then at about 10 hours, it stops spinning. So the first thing we did was that we developed a computational pipeline to sort of quantify the flow fields in 3D. And just very briefly, I will walk you through what we did. I mean, the way that we did it is that we just segmented the outer surface of this sphere. And then we do a projection, much like how we represent the Earth on 2D surfaces. We do this Mercator projection, where this horizontal line is essentially the equator, the North Pole, and the South Pole. And then when we get good enough signals and good enough segmentation, you get tissue flows that goes in the that looks like this, where we can actually have information about the flow field as well as information about the cell shape. So, and then once you have this plot, you can perform PIV to actually quantify the flow field. And just to point out structure, sometimes you see that occasionally there will be structures where here you see a counterclockwise and here you see a counterclockwise and here you see a clockwise vortex. And if you think about it, if you try to map this back on the circle, these two vortices are actually at the North Pole and the South Pole if you have a solid body rotation. But after what we can do is that we can actually, once we have the PIV field with the proper correction, we can map it back into 3D. And then with that, we can actually, with the flow fields, we can compute this rotation axis for the tissue. So now that we have this machinery to sort of quantify the dynamics of this spinning tissues, we can start to see what the experiments tell us. So what we found was that there are actually different rotational modes in our experiments. There are cases where you see a persistent rotation, meaning to say the axis of rotation remains more or less stable with very little axis angle change. You get cases of a rotation arrest, whereby it rotates persistently for some time and then it just spontaneously stop rotating. And then there are also cases where there actually is a drift in rotation axis, meaning to say it rotates along some axis, some orientation, and then it sort of drifts to a new position. And this period is punctuated by a decrease in rotation speed. And what is sort of puzzling is that this actually all happens within the same tissue culture. So in the same matrigel dome, we see this different type of behavior, and we wanted to ask what is the underlying biophysical mechanism. So and then the way that we address this is from a modeling perspective. By teaming up with the group of Ulisha, we developed an active vertex model on a spheres. So here we just model each cell as a polygons, and then we prescribe a polarity vector in each of the cell. The first equation tells us the force balance. So the frictional force at each of these vertices is balanced by the attraction force. So meaning to say each of this cell will want to crawl along the direction of this polarity vector. And on top of that, there's a term that tells us the mechanics of the system. So this energy term includes the area elasticity, which accounts for the pressure of the cell as well as the cell bond tension. And then we also need an equation to tell us the evolution of the polarity vector. So there's a term that says that the polarity vector wants to align with their neighbor with some rate gamma. And then there's noise term. And then these other terms are just terms that we need to keep everything on a 2D spherical surface. So what we have then is so if we initialize simulations whereby the polarity vector is all random, but then with sufficient alignment rate, what happens is that the polarity vector will very quickly align, and once they line up, they will perform this tissue rotation motion. So then the next thing we did was to ask, what are the different phases that we can get from this active vertex model? And so the two parameters that we tune is this alignment rate, so meaning to say how quickly this polarity vector is lining up, and the traction force, how fast the cells would want to move in the direction of the polarity vector. So in this regime where the traction force is relatively small with respect to alignment rate, we have this solid regime whereby the polarity vector very quickly line up with their neighbors so that you can perform a persistent global. Yeah.
Is there any planars of polarity?
So we tried looking at, so experimentally we tried looking at it. And then of course, in other systems like Drosophila, there is a PCP pathway, but I think that is on the basal side. There's a lot of literature on PCP pathway on the apical side, but we don't think that's we think we are looking for some PCP at the basal side because that's where the cell is crawling. But these are mouse stem cells which are hard to get in the first place, and we don't have the necessary cell line to label or perturb the pathway. So yeah. Okay, and then the other thing was that in other regions where your traction force is large relative to alignment rate, meaning to say, hand waving in an intuitive way means that the cell will move faster than it wants to line up with their neighbors. In this case, you will get a flowing regime whereby there are more chaotic motions, but then you don't see this global tissue rotation. So then so once we see this phase diagram, we wanted to ask, how does our experiments compare? Where in this phase space do our experimental states live in? And so we define this solid body rotation fraction, which basically quantifies the amplitude of solid body rotation with respect to the amplitude of all modes. And what we see is that in simulation in solid rotation, this F rotation is close to 1, while the flowing states has a small rotation fraction. And if we quantify the same rotation fraction in our experiments, we find that most of them are actually tends towards the solid rotation regime, meaning to say our systems lives close in the solid regime. What is interesting is that in our model, it also tells us that at the boundary between the solid and flowing regime, we actually have a yielding-like transition, meaning to say you have states where the tissue still spins, but then there's a random drift in the rotation axis. And this is, if we quantify, and this reminds us of this drift in rotation axis that we see in our experiments. And if we quantify the rotation axis orientation as well as their angular speed, we see some qualitative agreement. But we need to do more quantification to sort of make this comparison more convincing. And because of time, maybe I'll just briefly mention one last thing, which is that on top of flow field, we also have cell shape information. And this is interesting because the cell shape actually tells us something about the mechanical stress in the system. So with this information, we actually quantify this angle beta, which is the orientation of the cell elongation axis with respect to the local latitude. And so naively, you would expect that if the cell shape is completely random, the distribution of beta should be a uniform distribution. But to our surprise, what we found was that we are states in which the cells align up with a peak at 45 degrees, meaning to say they line up in this fashion. And then there are also states in which they line up in has a preferred angle of 135 degrees, which pictorially looks like this. And if you look at them, you will notice that these are actually a mirror image of one another, meaning to say macroscopically there's a chiral symmetry breaking that goes on. And just for control, we find that in spheres that do not rotate, this probability is flat. So to cut a long story short, I will just briefly mention our current understanding of where this chiral symmetry breaking is coming from. So we wrote a continuum model of this polarity vector. Basically, what this is that the dynamics of the polarity. There's an alignment term, and then there's a convection and rotation term, meaning to say so basically what this means is that our polar vectors are on the spheres, and as the spheres has some velocity field, it will convect and rotate the polarity vector. So what we found is that if you solve for this equation for symmetric steady state solution, there are azimuthally symmetric steady state solution, you sometimes will get states that looks like this, in which the cell orientation distribution will be bimodal. But then you also get chiral states in which the cell shape orientation actually has a preferred angle of 45 and 135. And the origin of this is that depending on the kind of defects that you have. So here, if you look at this defect here is actually a vortex defect. Whereas the defects at this point is actually a spiral Ester-like defect. And depending on where the defects are on North Pole or the South Pole, you actually get a twist in the polarity field, and this is the origin of the chiral symmetry breaking. So that's all I have to say for this story. So what we found basically is that polarity, the interplay between polarity, alignment, and traction force in a vertex model is sufficient to recapitulate the different rotational dynamics that we see in experiments. Experimentally, our spheres actually rotate close within this solid regime, and then the asymmetric displacement of topological defects actually tells us underlie this chiral symmetry breaking that we see in our system. So far I've told you dynamics of this type of a single epithelial structure. One thing that I didn't emphasize earlier is that the reason why we work with this stem cell organoid culture is because they actually come with different cell types and they are supposed to mimic organ development. So in our culture, we also see different types of structures such as the cuboidal spheres, compact ones with some branching morphogenesis, and also structures that has a lumen. So these are our hope is that we can use this system as a model for morphogenesis. And then we can ask questions like whether these cells actually exhibit polar or nematic order in 3D, and whether topological defects in this 3D actually determines the lumen formation. For example, whether you get a single lumen versus a multiple lumen. We are developing synthetic biology tools to try to manipulate some of this shape. So this is an optoshroom optogenetic cell line where we can activate apical constriction using light. As you can see here, when I turn on the light, the lumen actually constricts and become smaller. And then lastly, because of the different morphologies that we can get in our experiments, we are hoping to explore this idea of mapping the morphological landscapes of epithelial organoids. And the idea here is very similar to this transcriptomic landscapes that people have for RNA expressions, except that in that case, we are looking at gene expressions. Here, we are actually looking for morphological features and try to see if by mapping this morphospace, can we extract a tissue assembly rule. So these are the two main threads in my new lab. So one on chiral active matter, the other one on organoids using organoid to study morphogenesis. Of course, we are actively recruiting PhD and postdocs. So if you know of any friends or if you are interested in some of this, I'm available to chat. And then I want to thank these two groups of fantastic collaborators. On the first one, mostly at MIT, where we study living chiral crystals. The second group at Max Planck Institute in Dresden, where the work of a rotating organoid was done. I want to thank you for your attention, and then I'm happy to take questions.
The last part. So the lumen is fluid, right?
Yes.
So where's the traction force coming from?
Outside. So remember the basal side. The basal side is outwards. So it's like the cell is crawling. You have a sphere. The cell is crawling outwards, not inside. Yeah.
Can you relate the alignment parameter and the traction force to something physical and see that like maybe like in one case the traction force is less, the alignment parameter is less?
It's very hard to calibrate traction force and polarity. So the alignment rate. So with the polarity, we don't even know what the polarity system is. So we will not talk about the alignment rate. With the traction force, I mean, it's going to be a combination of I don't know how much integrins you have, etc. So I feel like the best that we can do is that we can have drugs that tune the attachment rate, for example, so that we have one level down and one level up to see if we can push the system into the fluid system.
So, like you said that in the same batch for all the three modes.
Yeah.
So it's just like random variations of this parameter space.
Yes. Yeah. So then so you're right. There's some level of variation even within a system. So then we have to talk about the distribution of solid versus fluid as we go through the different perturbation.
In the dynamics you wrote down for the polarity, but for the vertex column, is that just an orientation or is there an amplitude? Like how
yeah, so we cheated a bit. We did not model the velocity as well. So in the continuum model, the velocity is imposed. But the point that we were trying to make there was just to say that in the velocity field that we are interested in, you get chiral symmetry breaking. But we are not trying to recapitulate. But you're right, the full model will include the velocity, and then it becomes complicated.
So from my understanding, like the vertex model is the polar model.
The vertex model, yes.
That I think directly the fact
So the Hairy-Ball theorem tells us that you must have a net +2 charge, but you can have many more defects, plus and minuses, so long as the sum is +2. Yes.
Or like,
Yes. Yes, that's right.
then we consider like, I'm not sure if this is true that the polar factors, or if this is like hexagonal symmetry, like experience when you
Right. So we haven't explored the hexagonal order of the cell shape. So the cell shape and the polarity are two different things. So that's the first thing I want to say. We actually assume that the cell has some nematic order. So basically we fit an ellipse and then we find the elongation axis. But you're right that there could be higher order, like hexatic order. And in fact, I mean, the group of Luca Giomi and others are looking into that, and there's already a couple of papers on that.
Yeah, my point is that probably there's more, much more defects, and then much more defects.
Yes. Yeah.
So when you look at this outer epithelium,
yeah,
do you see also intercalation, or the neighbors of each cell will stay the same over time?
So we try to quantify that, and in fact, that is actually the right definition for solid rotation, in which you have very few neighbor exchange, and then in fluid states you have a lot of neighbor exchange. So what I can say is that we try to quantify that. It's very difficult because you have to segment things, right, and things like that. But then by eye, it looks like it's definitely more on the few neighbor exchange versus yeah.
So the vertex model, does it implement intercalation or not in your simulation?
Yes. So in the simulation, in the fluid regime, you do see a neighbor exchange. Yeah. Other questions?
Are there any conditions when your model has the kind of like random flowing phase? Is there any conditions when you see that in an experiment?
So in our experiments, I think if you look at large enough spheres, we actually see some indication of that. Because so here we try to keep things simple by just focusing on a certain spherical size. But then once there's some correlation length in the model, and if your system size is bigger than the correlation size, then you naturally get this flowing states. But then experimentally, it's hard because once you have this big organoids, then they start to fuse with their neighbors, and then you get dumbbells and more complex shapes. And yeah.
And then also, can you had to like stop rotating?
Yeah.
That you observed in the experiment. Is
right.
there any way you're like to model that?
Yeah. So in a model, there are sort of two ways. Well, more than that. You can turn off traction. You can turn off alignment. Both will stop rotation, but they actually leave different signature in terms of the cell shape. Because if you turn off alignment, the polarity vectors becomes random. The system is able to relax. But if you stop traction force, then the cells will get into a mechanically frustrated states. And actually, we try to quantify a bit of that in experiments, and the indication is that it's actually turning off of traction force and not turning off of polarity alignment.
If there are no more questions, let's then.
Okay. Okay.
Maybe we can discuss this tomorrow.